Welcome, everyone. Welcome those of you who are here for the first time. Welcome those of you who are repeat attendees of Within Sight. Ooh, the microphone just got louder. Welcome to our judges, and of course, welcome to our 2016 Image Award winners and their family and friends. I am Anna de Koning. I'm the Executive Director here at the Koch Institute, and I'm so glad you're here today to share what is one of my favorite days of the year, namely the unveiling of the new images. Every year anew, these images offer glimpses into the wondrous world of research and really get at what drives KI researchers, which is the promise of scientific and technological advances converging to overcome disease. I think the images really speak for themselves in showcasing how amazing the work here is at the Koch Institute, and I really want you to know how honored I am to be here. The whole image competition, the judging process, the installation, the maintenance of the gallery, frequent Koch Institute tours, school visits, this evening's event, the whole Within Sight program are all thanks to the hard work of our Koch Institute Public Outreach Coordinator, Erika Reinfeldt. So this evening and all the credit belongs to her. And so I hope you have a wonderful evening, and I am now passing it over to Erika, who will MC the night. Thank you, Erika.
Thank you, Anna. I don't think all the credit belongs to me. I think there are some fabulous people in the crowd here who actually created these images and supported the people who made them. But I am thrilled to be the MC for this event. It is also one of my favorite nights of the year. And I hope everyone out there, first of all, let me check in. We had some work done on our audio system this morning. Can people hear me, especially in the back? Yeah, excellent. Thumbs up all around. All right. Well, I hope that continues. All right. So the way this is going to work is I'm going to moderate the discussion, and we've got 9 presenters, each of whom are going to present their image in what we hope will be 3 minutes or so. And then at the end, we'll save all questions to happen out in the galleries in front of the images. So if you have questions that you want to know more about, we invite you to accost people afterwards in the galleries over dessert. So my job tonight is to facilitate and make transitions between the various stories, because the images are chosen individually and then together they become the exhibition. But of course that wouldn't happen without the support of some people that we absolutely need to thank. These images are not chosen by me and Anna. They are chosen by a wonderful panel of judges. And I think we have some of the judges here. Could you all stand up, those of you who are here? All right. I know at least one person went into. Hello, Deborah. All right. So yes, I got an email from, I think it was Mary, saying she got the flu that's been going around. We also lost a PI to that. So we're especially glad that all of you are here and healthy. But our judges spend about 2.5 hours deliberating over the images. They narrow, this year, more than 150 images down to a short list. And then from that short list, they choose the 10 winning images. So it is exciting, and there are always a few images that we wish we could put on display, but we've only got 10, and we've only got an hour, so let's jump right into it. All right. So our themes for the evening, Anna mentioned the promise of scientific and technological advancement. When we asked our judges to choose images, we said to them, here are some themes to keep in mind. We asked people to consider these when they were writing up their submissions, talking about Mind in Hand, the spirit of MIT. We're celebrating MIT's 1st century in Cambridge. We're also celebrating the 5th anniversary of the Koch Institute Building and Public Galleries, which brings together, sorry? I thought I heard I can't hear you. All right. Here, got it. See, I'm very sensitive to words that sound like other words. You're going to notice that later. All right, so this is our 5th anniversary of bringing biologists and engineers together under one roof. And also, we are celebrating 5 of the pillars of progress, which you can see on the right here, with an eye towards cancer prevention. So I'll try to highlight a few of these throughout the evening, but I promise this is the last text-heavy slide you're going to see. What I want to do is jump right in and start with the image that I think you've actually been staring at for quite a while, which is the image on your program. This is a complex image, but it beautifully represents the diversity of the exhibition as a whole. Understanding its content and its creation requires knowledge of cell biology, materials chemistry, even robotics. Suit Your Cell, like choosing an outfit, it's kind of difficult to decide which story we want to tell about it. Luckily, that responsibility falls to Asha and not to me. So I'm going to go ahead and invite her up to tell a tale or two about the image.
So my name's Asha Patel. I'm a postdoc in the... I'm a postdoc in the Anderson and Langer labs. And this image here represents a quest for new materials for the growth of human stem cells and their derivatives. So each image is actually a compilation of separate studies using specific cell types, such as stem cells, skin cells, heart cells, their nuclei, and the last one is my favorite, is a material which we couldn't actually use because it had so much background fluorescence. So that one was actually a useless material. So why do we want to culture these cells? Well, stem cells reside in various places in our body, and their job is to maintain and regenerate damaged tissue. And they can also self-renew to maintain that regenerative capacity. For example, we have limbal stem cells in our eye that are constantly working to regenerate damaged cornea. And also now we can turn skin cells into stem cell-like cells, and they can turn into any cell type in the body. So for these reasons, these cells are extremely valuable for providing disease models of hard-to-access tissue like the brain and the heart. And they're also useful for cell therapy, and bone marrow actually is one of the oldest forms of cell therapy, whereby the stem cells inside bone marrow can turn into blood cells. So for wherever these cells come from and for whichever application we need them for, we need robust culture systems where we can grow them outside of the body. And so that was the focus of our screening experiment, was to look for materials, bespoke materials that we could use to tailor-make systems for specific cell types. And we particularly wanted synthetic, non-biological materials that could be made to a consistent standard. So how do we develop these materials? Well, we don't know enough about how stem cells interact with synthetic materials to enable a rational design approach. And so we effectively provide a wardrobe of materials to our cells, which they are free to choose from. So you can see here the materials are printed onto a glass slide, and then cells are placed onto this glass slide, which they are free to interact with the materials they prefer. So you can see on some of the spots they've attached, and on some spots they haven't. And in this example, we've got heart cells. And we then correlate this to the exact chemical composition of each material, which we know from detailed characterization. So once we have a material we've identified, we then really focus in on whether these cells are functional on these materials. And you can see when we zoom right into each spot, there's a whole depth of information in each image. So in the case of heart cells, we specifically stain for a cardiac protein, which is found in the cardiac muscle fibers called alpha-actinin. And if this protein is banded parallel to each other, it means that the fibers are well aligned and they're more likely to produce functional cardiomyocytes that can beat. And so on this material, if you focus on the video, it's actually beating. Don't blink or you might miss it. But this material, the cells are actually able to beat, and that's because they have a good organized structure. And so we took this material forward into a drug screening assay where we were looking for the effects of an anti-cancer drug, doxorubicin, which is known to disrupt these myofibrils or muscle fibers. And so we found that this material gave much more consistent results than a biological system material. So we believe we are just a heartbeat away from using all the information we've gained from these screens and to rationally design new materials for specific cell types. And so I've only had the chance to talk about the heart cells in this study, but there were many cells that we worked with. So if you'd like to speak to me afterwards, then you can, or if you want to hear more puns, which I have plenty of. I had one on each slide for those of you who noticed, and one on my final slide. So I'd like to thank an array of people who made this work possible, in particular Morgan Alexander and Chris Denning from the University of Nottingham in the UK, who have materials background and stem cell expertise, and at the Koch Institute, the Anderson and Langer Labs, who really pioneered the robotic printing technique. And my funding body, the Engineering and Physical Sciences Research Council, and the Koch Institute for giving us a platform to not only do brilliant science, but to showcase our art as well. So thank you.
Thank you, Asha. I'm excited about this project. I think it really has the potential to address a lot of things. There are going to be a lot of them. Settle in. All right. So this idea of using using synthetic materials, engineered materials, to understand principles of biology is, in fact, the theme of our next image. So Russell's here, so let's just move on ahead.
Thank you. My name is Russell McConnell. I'm a postdoc in Frank Gertler's laboratory. And the image that the judges selected is shown here. And these are, I'll call them neural-like cells. So these are actually derived from a neural tumor, and I'm using them as a model system to study neural development. And when I say neural-like, they're not really neurons because they're tumor cells. They're cancer cells. They don't behave normally. So the cells that I would like to study, that I study all the time, are shown here in an image not selected by the judges. So these are the dorsal root ganglia cells, is their technical name. But basically what these neurons do is the neuronal cell bodies are these sort of circular yellow things that you're seeing here. And what they do is connect receptors located all throughout your body, basically up to your head, so you can perceive different sensations. So when you touch something, these are the neurons that are carrying a lot of that signal from your fingers all the way up to your brain. So as you might imagine, this connection has to be quite long. So how do they do this? So when we take those cells that I showed you in that first image and we plate them on glass or plastic or hard substances like we typically do in the lab, they basically just spread out. They form these sort of fried egg-looking morphologies. They just sort of flatten out. But if you plate these cells on a very soft hydrogel, they do something quite interesting. So they don't flatten out, they stay rounded up, and then over time they actually extend these very long processes. So instead of looking like this fried egg shape that we see up here, they look something that looks like a neuron. So here on the right side, sort of in the pinkish color, is the cell body, and this green line that's going across the screen is an axon. So this is actually what neurons use to connect different parts of the body together. So what's really interesting about this is that the cells are sensing the mechanical environment, and that's informing their decision about the shapes that they're going to make. So what we're really interested in studying is how does this mechanical cue influence the cell's behavior. So under normal conditions, a healthy human body doesn't have a lot of glass or plastic in it. So we're really using these mechanical materials to mimic a natural environment to elicit these normal behaviors out of these cells. So when you look at groups of cells, on the left, these are the same cells cultured under identical conditions, the only difference being the cells on the left are on a glass slide and they're just linked together, whereas the cells on the right are on this soft gel, which mimics their natural environment, and they're sending out these long processes, which they can then use to make connections to other cell types. And I was going for the record with the fastest talk, and I think I managed it. So if anybody has questions, I have no puns or acknowledgements, but feel free to talk to me afterwards. Thank you.
Thank you, Russell. It really sounds like you're asking some good questions. All right, sorry, we had to put one in there. All right, so what I want to do with this next image is actually to go back to this idea of a 3D matrix to study biological phenomena. This is our image from the Wellcome Trust. This comes to us by way of London. We have an exchange program with Wellcome Images in London. Every year that we've mounted an exhibition, we've included one of their images, and every year, and since then, they have also included one of ours. So this is a stem cell embedded in a porous hydrogel matrix. Over the years, we've showcased plant cells, zebrafish, bacteria, and now the stem cell. And I couldn't resist this title because, as Anna mentioned, in addition to running the Image Awards and the exhibitions and public gallery events, I also run our middle and high school field trip program. So I'm a big, big advocate of STEM education. And I mention that partly to say if you know any middle and high school students who would like to come visit us, we'd be happy to show them around. But mostly I mention it because I want to use the excuse to tell you that the next image we're going to talk about is absolutely, despite its title, not child's play. The original title for this that we came up with was actually Global Understanding, but I think it was Dan's wife. Dexter's wife, sorry. Dexter's wife who came up with this much, much better title. And I like it so much better because it is, in fact, a project all about how structures develop and what happens when things go afoul.
Thanks, Erica. So, hi everybody, my name is Dexter Jin, and I'm a graduate student in Piyush Gupta's lab, and I'm excited to tell you about this image that Daniel Miller, another graduate student, and I submitted this year for the image contest. Now, those of you who were here last year got a glimpse into a novel system that our lab developed to grow mammary tissue directly from patients in these hydrogels. And one of the amazing things that came out of this was the ability to study mammary development in a structurally maintained manner. Our lab showed that we could faithfully mimic normal mammary glands and use this to gain insight into how the normal develops. Now, early during development, we could see these tube-like extensions here called ducts, which later go on to give rise to balls of cells called lobes at their ends. Now, on the left, I'm showing an example of a more developed structure where here you can see purple marks all the cells in the structure. And in this image, you can see this tube-like structure along here, this axis. And at this stage, you can also see that the duct terminates with these lobes. Now, in contrast, what I'm showing you on the right is a structure that results when we specifically block a single gene. Three immediately notable attributes of this include the reduced number of lobes, the fact that the ducts are actually thinner, and that the lobes are misplaced. Instead of being more closely associated with the end of the duct, they're actually more closely associated with this mass of cells here. Now, academically speaking, this is a very intriguing result because we know that these lobes act as locales for milk production in humans, and that these ducts act as routes for the milk to travel through. So using this system, we were able to gain insight into how this gene in particular contributes to mammary development. But with the system, we can really do more than follow structural changes. We can also learn about which cell types are being affected by our disruptions by color labeling specific genes which are present only in specific cell types. Now, once you overlay this information, shown here in blue, you really get a sense of, one, where specific cell types are spatially oriented in the structure, and two, what fraction of these tissues are comprised of that specific cell type. Moreover, when we combine this with the inhibition of the genes that I mentioned before, we can see how these characteristics are ultimately changed. And when we put all of this together, we really get insight into how genes ultimately contribute to the overabundance or lack of production of cells that are important for processes such as milk production, or may contribute to diseases such as cancer. And with that, I want to thank all of the Gupta Lab, Piyush for all his support, Dan and Ethan for their work in establishing this amazing system, Dan especially for his equal contributions to this particular image, and the rest of the Gupta Lab, Yushang, Daphne, Rob, and Minu. I'd also like to thank the Keck Imaging Facility, in particular Wendy Salmon. Thanks.
Thank you, Dexter. I think your tale of two structures really showed us the best of times and the worst of times. Sorry, I don't want to pile it on too much. I know that is just going to annoy the dickens out of you, but I will just say that we have great expectations for this research. And actually, this research was just, an aspect of this was just published. And so I want to say congratulations to that. And I actually forgot to congratulate Russell and Frank on their paper being accepted for publication soon, as well as the next image that's coming up. It's almost like they planned it to match this. But I really actually wasn't joking about this idea of great expectations. One of the themes that I mentioned tonight is this idea of cancer prevention. And it's becoming very clear that one of the best ways to control this is that the exploratory, discovery-based research is what is going to lead the way for understanding the root causes of the disease and stopping it in its tracks, sorry. Or in the case of this next image, stopping it in the GI tract.
Thank you, Erica. Good evening, everybody. My name is Jatin Roper. I have a bit of a different background than the other speakers. I'm a physician at Tufts Medical Center, specializing in gastroenterology, and I'm also a scientist in the Yilmaz lab here at the Koch Institute. One of the common conditions that I treat in my clinical practice...
I forgot we're going to open.
Oh, yeah, thank you.
Yes. Jatin has a movie that doesn't play beautifully in PowerPoint.
Great, thank you.
There we go.
Thanks, Erica. One of the common conditions that I diagnose in my practice is colon cancer, unfortunately. Colorectal cancer is the third leading cause of cancer mortality, and the lifetime risk of developing colon cancer is 6%. In the lab, we are interested in learning how conditions like diet and aging affect cancer formation in the colon. One way that we study colon cancer in the lab is to grow intestinal tissues in culture as mini intestines, also known as organoids. We then use these cultures to understand how normal intestine develops into cancers under certain conditions. However, it is unclear how these dish-grown structures respond to the actual living conditions that are located inside the body. And so therefore, we have developed a method to transplant these organoids into the colons of recipient mice using colonoscopy. So in the video I'm going to show you, you're looking here at a mouse colon during colonoscopy. The mouse is, of course, sedated and very comfortable. And what we do is we place a needle, shown up top, directly into the wall of the colon. And then we inject the organoids into the wall, after which the organoids then stick into the host mouse colon. So that's the transplantation process. Now, to confirm that these organoids successfully engraft and are living inside the recipient mouse colon, we then take a look at a slice of the recipient colon a few weeks later. And what we see in our image in green and blue is the host colon. And just below it, in pink, you see the transplanted organoids. And so this image provides evidence that we are able to derive intestinal cultures, also called organoids, from one mouse, and then successfully transplant them into another mouse colon. The significance of this research is that we are able to answer biological questions that we could not answer previously. In current experiments ongoing in the lab, we are modifying the physiological conditions of the transplanted organoids as well as the conditions of the recipient mouse. We are then using this approach to study how conditions such as caloric restriction, high-fat diet, and aging affect cancer formation. And we hope that our findings from these studies will one day lead to novel therapies for colon cancer. Thank you very much. And I just want to close by thanking my close collaborator, Tomas Tamela, who's in the audience today, as well as our mentors, Omer Yilmaz and Tyler Jacks, our grant support, and in particular, the support of the Microscopy Core Facility and the Hope Babette Tang Histology Core Facility. Thanks again.
Incidentally, a paper on high-fat diets and cancer risk and initiation was actually the spotlight on MIT News today. We didn't even pay them to do that. But I'm really worried. Okay, we just went back to the beginning. All right. Let's see who remembers what. All right. And I told Jatin we would keep his presentation away from the food on either side, but if anyone has more questions about that, I'm sure he and Omer, if he's still here, would be happy to talk with you about high-fat diets and cancer over dessert. Now you know why we needed presentations to stay short. All right. There we go. All right. So here we go. All right, so what I want to do now is switch gears and talk about another aspect of cancer prevention, particularly early detection. As anyone who has come up against cancer knows, it's tough to find and difficult to remove. So our next presenter, Li Gu, is going to talk specifically about how he and his collaborators are working to find tiny early-stage tumors. But as you'll see, this collaborative project also presents a wide spectrum of strategies for treatment as well.
Thanks, Erica, for the introduction. Hello, everyone. My name is Li Gu. My name is Li Gu. I'm the postdoc in Hammond Lab. I'm here to represent myself and Dr. Xiangnan Dang from Belcher Lab to present this winning image. The title of the image is Beyond Red, means we're detection tumor in new light. So I'm going to give a little bit of background about light. So here's the electromagnetic spectrum of light. So going from left to right, increasing the wavelength, you have UV visible, near infrared, and infrared. So the near infrared is divided into near infrared region 1 and region 2. And I'm going to give you an example of the comparison of different light detection methods. So we're focusing on the near infrared 2. So for the past decades, people are using the visible and near infrared 1 to use for detection cancer, for the cancer detection. Now for the recent years, near infrared 2 is coming up. So here's an example of a abdominal region of tumor, and it's apparent that the near infrared 2 can pick up a signal with a much higher resolution. And if you look at the visible image, it's barely see anything. But if you look at near infrared 2, you will see that they can pick up some tiny tumor that near infrared 1 can do that. So with this collaboration in the Hammond Lab, we're developing a nanotechnology called layer by layer. So pretty much in this process, we put the near IR2 material into the core of the nanoparticle. Then we have modular layers in between. This can serve as a reservoir for future drug encapsulation. Then we have an outer layer for the purpose of navigation to tumor to reduce the side effect. So here's the winning image. So in the green spots are all the near infrared 2 nanoparticle. And here, which is a piece of healthy liver and a piece of healthy liver. And this is a small piece of tumor resting on the liver. You will see majority of the nanoparticle are getting into the tumor. But I will give you more examples that alone submit to the image award with the winning image. So here is the piece of gut, and this is a piece of tumor resting on the gut. You will see all the particles are in the tumor instead of the liver. And another example, this is a piece of tumor resting on the healthy pancreas. You see most of them, they are in the tumor. And another example is an interesting invasion of tumor inside the liver. And even the tumor area is inside the liver, you will see some particles are still able to travel all the way to the tumor. And this is an example showing that nanoparticles are spreading all over the tumor area. So with that, I would like to give acknowledgement. Me and Shauna want to thank our advisors, Paula Hammond and Angie Belcher, for their support during the work, and also the core facilitators. Especially want to thank Jeff Wyckoff. His 2-photon microscope was able to detect our material, because as you see that our material only emit in the near 2 light, which is invisible, and also the funding sources.
Thank you, Lee. Yeah, these presentations often end up being a, look, here are the other images that I submitted that the judges didn't pick. I remember this selection process. They spent a lot of time, they loved this story, right? So the criteria that we give to judges is we want you to pick images that are visually stunning and scientifically compelling. And so this was one where they loved the story and spent a very long time debating which image they liked best visually. And I think it was the contrasts between the angles and the curves and the bright and the dark that won the winning image over. And one thing that I wanted to note about this image is that I learned a new word when I was creating the caption. It didn't make it into the caption, but it is a great portmanteau. It's theranostic, which is a combination of therapeutic and diagnostics. And I think that is just very powerful, that, this particle can be created that can both diagnose and treat these cancers. And I'm also going to use that as my transition into our next image, which is a different engineering project to study something completely different, but it is also used for treatment and also to collect information about the process. So Jonathan Sosi, you're up next, and this is, he originally wanted to tell you a little bit about the technological history and how this is a project 70 years in the making, but I think he's going to, right, the point I wanted to make there is if you happen to be a technology history buff, you should find Jonathan afterwards, because he has a lot to say about the origins of this. Right.
All right. So good afternoon. I'm Jonathan Sosi. I'm a technical assistant with the Anderson and Langer Lab. All right. So actually, all right, so restorative bionics was first popularized by the 1970s TV show, The $6 Million Man, a TV show about a NASA astronaut that had his injured limbs and eye replaced by electronic implants. However, the reality of such implants has been hindered by the rigid structure of silicon. Silicon was first implemented in the transistor in 1947 and has since become the basis of all electronics. So the focus of our research is to re-engineer electronics to better integrate with the soft tissue of the human body. And there are 3 major approaches that we have developed to do this. And the first approach is to get rid of silicon altogether and to print these electronics onto a stretchable platform, such as the one shown here. And in addition to providing a printing platform, it also provides electrical insulation. The second technique is to actually take electric components and put them side by side in a parallel format to allow them to conform to curved surfaces, as shown here. And the third approach is inspired by nature, by taking these patterns and redesigning electrical circuits to allow them to stretch in multi-directions. And so right here we see a Pinot design, which is a fractal pattern, which allows us to stretch these hard metal circuits in multiple directions. All right, in 2011, Congress approved $50 million for the Joint Warfighter Medical Research Program, a program aimed to develop technologies for combat-related injuries. Over the past decade, hundreds of thousands of soldiers have sustained injuries that have led to peripheral nerve damage, a condition in which the nerves that send messages from the brain to the muscle have been severed, causing paralysis and atrophy. So as part of this program, we have developed a stretchable implantable microelectrode device that electronically stimulates the muscle during nerve repair, promoting contraction and preventing atrophy and drastically improving rehabilitation time. And in addition to providing stimulation, this device records the neural process during nerve repair, supporting ongoing research in neuroscience. Right, so one of the wonderful things about electronics is that we're able to program them. So we're programming wireless capability into our device using RFDuino and stretchable antenna design, allowing us to make something that looks like this do the same thing as something that looks like this. And because we are the Langer Lab and the Anderson Lab at MIT, we're always pushing the envelope of technology. And so we're developing technology and using new materials, such as the one that I'm about to show here, called Gallistan. So basically, this is a liquid, a metal alloy, which is liquid at room temperature and has been used as a substitute in thermometers. And basically, the idea is to implement this liquid alloy and put them into channels like this to make flexible electronics, pretty much like the cell phone screen, making capacitive touchscreen electronics that we could possibly embed and use as therapeutic devices for diseases. And I want to thank our team. So our electronics expert, Dr. Span Adauki, and our wonderful mentor, Dr. Khan, Kurt Broderick at the MTL, who's helped tremendously in electronics design, Mr. Bavois for putting me in and helping us take images to allow us to win this award, and Dr. Anderson and Dr. Langer, and also the Department of Defense and the Koch Institute for putting on this event. Okay.
Thank you, Jonathan. I think this image is actually the one that has the most puns in the caption itself. So I'll spare you them for this. What I want to do now is continue on our engineering-heavy portion of the evening. This happens to be another regenerative medicine project, and it's one of two presentations about work being done in the laboratory of Sangeeta Bhatia on the 4th floor in this building. Sangeeta herself is off running a student science fair tonight, so she can't be here, but luckily Chelsea has agreed to deliver a quick summary of what the lab does.
All right, hi everyone. So I'm Chelsea Fortin, and when this image was taken, I was a technical assistant in the Bhatia lab. I'm now at the University of Washington, but I'm really excited to be back. So I'd like to talk to you a little bit about this image. And so liver disease is one of the two facets that the Bhatia lab explores. We explore cancer, which you're going to hear a little bit from Alex in a couple of minutes about our cancer work. But our team also works on liver disease. So this is a world map of the liver disease death rate. So you can see this is a very significant world health problem. In the United States, around 36,000 people die every year from liver disease. And worldwide, 1.3 billion people are living with liver disease and its complications. So I'm going to tell you a little bit about how we've tried to engineer a solution to this. So first, we take liver cells, which I'll refer to as hepatocytes throughout this presentation because I'm just so used to that technical term. So our hepatocytes, we pattern using similar computer technologies that were just mentioned with silicon wafers. We pattern these hepatocytes into these ball-like structures. And those ball-like structures, which we call aggregates, allow the hepatocytes to function much more physiologically outside of the body and in an in vitro system. And then we also pattern cords of blood vessel cells by using similar micropatterning technologies. And the whole idea is that we're trying to mimic the physiologic environment and architecture of the liver. So by combining this vasculature component and our aggregates of liver cells, we are able to form an implantable liver satellite, which is implanted into the abdominal space of the model, but not necessarily near the liver, because by the time a liver reaches failure, it's a very inhospitable environment, and if you tried to inject healthy cells there, they won't be able to function. So here is an image of this implant under the microscope. This is what it looks like when we implant it. It's about 6 millimeters in diameter. And under the microscope, you can see, again, our cords of blood vessel cells in green and our balls of liver cells in red. And I'd like to invite you to think about one of these balls of liver cells as we examine the winning image tonight. So one of those balls of liver cells has now, after about 3 months of liver injury, become this red structure here. So this entire image is actually one of these balls of cells. So we're able to harness the liver's own regenerative function and get it to expand our implantable tissue. And in green, we have here our human blood vessels that we implanted as well. And you can see similarly, they're no longer in a cord-like structure. They've organized themselves. They are perfused with white mouse blood, which is very exciting because it's in vascularization of complex tissues is one of the largest hurdles for bioengineers and tissue engineers to overcome. So with that, I would like to thank the Bhatia Lab, the LMRT, specifically Sangeeta Bhatia for all of her support, and Kelly Stevens, who is my direct supervisor. And then I'd also like to thank the entire Koch Institute, but especially the microscopy and histology cores, because without them, we couldn't have done any of this work. So thank you very much.
All right. Thank you, Chelsea. I realized this was another candidate for the title of Lobo Understanding, so that's a bloody shame that we missed that. But speaking of blood, I'm actually really excited that you chose white to represent the blood cells, because our next image that we're going to talk about is actually inside of white blood cells. And like the Satellite Liver Project, it is a story of leveraging the body's own resources to fight disease. In this case, it's cause cancer. In this case, it's fighting cancer. And so let's do this because we are hungry like the macrophage. Ali, come on up.
Good evening. My name is Ali Roganian. I'm a visiting scientist here from the University of Southampton in the UK. So I am going to tell you about a bit about the background of my research. So I'm a cancer immunotherapist, and this picture explains to you why we need to understand the immunology and the body's immune system in order to fight cancer. As you can see here in this diagram, there's a tumor. But if you examine the tumor microenvironment, what you'll appreciate is that, in fact, a majority of the tumor bulk is non-tumor cells. And a lot of these fall into the immune cell category. So a lot of the white immune cells are, in fact, embedded within the tumor microenvironment. And that's why we believe that using these immune cells would be a great asset to fight the cancer. And in fact, in recent years, cancer immunotherapy has reached a new stage in the cancer therapy platform. And following the exploitation of this field, there have been a few trials which have managed to treat certain cancers, such as melanoma, skin cancer, and lung cancer, that in the past were almost incurable. And because of that, the prestigious Science Journal in 2003 named cancer immunotherapy as the breakthrough of the year. So in particular, we're interested in antibodies. Monoclonal antibodies are Y-shaped proteins, which are released normally in response to infection. So if you're infected with bacteria or viruses, your body's immune cells, the white blood cells, would secrete these antibodies, which will recognize the invading pathogens, and in turn, they're cleared by the phagocytes or macrophages within the system. And this is exactly what we've done here. So what you can see in orange are the cancer cells, and what we did here was to flag them up to the immune cells, which are depicted in blue here. So we have treated the cancer cells with a monoclonal antibody, which is in fact used in the clinic. And when we introduce them to the macrophages, macrophages have actually started engulfing them. And you can see one single macrophage shown in blue here can engulf or eat up to about 10, 20 of these cancer cells at a time, which is quite fascinating. But what we need to do is go beyond that, because cancer cells also have ways of evading the immune system. And a lot of what the current research is focusing on is moving beyond the current therapy and trying to make this a 100% kind of cure rather than a temporary cure. And what we're basically trying to do is to get all of these cancer cells, which are still left outside the macrophages, and remove them from the cancer, from the body. And that, whoops, what happens there?
Your acknowledgements have disappeared. We can.
No, okay, well, that brings me to the end of my presentation, and I was going to. Sure, I was going to thank the supervisors involved with this project. So Zhenju Chen here at the Koch Institute, and Professor Mark Cragg at the University of Southampton, and the scientists who helped with this project were Sonia James and a graduate student Gesa Sogriato. So if that comes up. And there you are. And the funding body is from Bloodwise, which is a UK charity fighting leukemia and lymphoma. Thanks for your attention, and thanks to the judges.
All right, I'm sorry about that. All right. And let's see if I've learned my lesson about starting and not going through all the presentation again. All right. So actually, while we're transitioning here, I did want to note that our next With Insight event is all about cancer immunology. That'll be on May 25th. It's on the back of your program. So Tyler Jacks, our director, will be presenting a master class about some of the cancer immunotherapy work that's happening here. And then a couple weeks later, our annual summer symposium is on the theme of cancer prevention. So those are some upcoming events that I thought you'd like to know about. But I realized that we are but one presentation away from dessert. So, all right. So we're going to move on to our next image, which Alex Albanese, also of the Bhatia Lab, is going to present. I'm going to give you another glimpse behind the scenes. We originally, when Alex and I sat down to talk about this image, we wanted to call it Close Encounters of the Third Dimension. But that was too long for a main title and too vague for a subtitle. And so not having both just messed with my aesthetic. And so it was just flat out, as was my Shakespearean approach of 2D or not 2D. So. No, you laugh, but it is important because this image is actually coming back to that earlier theme. It brings it full circle, if you will, about using three-dimensional matrices to improve our understanding of how nature works. And it also happens to address the one focus area, cancer research focus area in our building that I haven't yet talked about, which is metastasis and the spread of cancer. So as Chelsea promised / warned us, Alex is going to take a look at improving the design of nanoparticles to find and fight cancer.
Is there a laser pointer? Laser pointer? No, no worries. It's fine. It's fine. Got it. All right. Can everyone hear me? All right. So my name is Alex Albanese. I'm a postdoc at MIT, and this is work I did while I was at the Bhatia Lab here at the Koch Institute. And this image that's hanging in the gallery right now is an image of 3 cancer cell clusters in a network of proteins called collagen. And I'll get into why we took this image. So what underlies this image is my desire to design better nanomaterials. So nanoparticles are tiny little particles, as the name implies, that can be administered into the body. And the goal is to come up with the right formulation that allows us to maximize accumulation in the tumor. So we have a virtually infinite number of designs, and we're trying to figure out the best one that can allow us to deliver contrast agents and therapeutics into the tumor. And the only way we can do this is to really understand how the body interacts with nanoparticles. So during my PhD, I spent a lot of time looking at how nanoparticles travel through the tumor environment. And one thing I realized is they end up spending quite a bit of time navigating through the stuff in between the cells before they finally reach cancer cells. Now, the space in between cells is not empty. It's filled with a dense network of proteins called the extracellular matrix, or matrix for short. And one of the interesting properties of the matrix is that cancer cells actually have a very unique matrix relative to healthy tissues. So coming to MIT, I want to develop nanomaterials that can target this interaction between cancer cells and the matrix. So in order to do that, I have to start to understand how cancer cells actually interact and modify the matrix surrounding them. So to do this, we did a series of pilot experiments where we took different cancer cell lines and we grew them into these little clusters of cells. We call them spheroids because they kind of look like spheres sometimes. And we took these clusters and we laid them on top of a collagen gel. And the reason we chose collagen is 2 reasons. One, it's the most abundant protein in the body. It's in all of our tissues, in our kneecaps, ligaments, so on and so forth. And the second reason is collagen has this really interesting property that we can actually see it without using any fluorescent labels. So all the white strands we see here are collagen fibers, unlabeled as is. In these experiments, the cells are labeled in blue. And what was apparent from these experiments is depending on the cell type, each one interacts with the matrix very differently. So on the left, we have 2 cancer cell lines here derived from the JAX lab in the Koch. And these lung cancer cell lines interact with the matrix. They bind and they reorganize very slightly the collagen around them. And on the right, we have pretty much a very striking reorganization of the collagen matrix into these beautiful lines. And these are mammary cells, and they're able to connect one cluster of cells to another cluster of cells by these long collagen lines. The image that hangs in the gallery is an interesting line of metastatic breast cancer cells that when we generate these clusters, they have a layer of protein that surrounds them. So we have these 3 clusters, and they're embedded in the collagen matrix here, and it looks like there's a gap, but there's actually protein that fills this gap. It's just that we can't see them using this imaging technology. So when you see this image hanging in the gallery, I want you to remember 2 things. 1, this gap is not actually empty, it's filled with proteins. And 2, more importantly, I want to emphasize how important it is to understand how cancer cells interact with their environment in order to develop better nanomaterials and therapeutics for patient care. So this work was not possible just by myself. I'd like to thank Sangeeta Bhatia for a chance to work in her lab and to do this exciting work. I also want to thank Jeffrey Wyckoff. I don't know if he's here, if he's attending. Oh, he's back there. He's part of the Koch microscope core. Everyone who's worked with him knows how great he is. This image, interestingly enough, was taken in my first day of training on the microscope, so it's at the, the bar really high for future images. And I'd also like to thank Justin, Ben, and Arnav for productive discussions that led to optimization of these experiments, and my funding sources. And last but not least, it's been a real privilege to share my science with you guys today. And if you enjoy these types of conversations of scientists discussing their work with the general public in an informal setting, I encourage you to check out glimpse.mit.edu. It's a series of interviews that we're doing with researchers across MIT campus. And I'm actually going to try to interview some of the people that presented today. So if you want to learn more about their work, check out this website. Thank you for your time. And I think we have some desserts that are promised to us, right?
We do. Thank you, Alex. All right. Although I will say that before our presenters leave, at least, I do want them all to come up for a big group photo. And I would also invite any of the people who were named on the slides who are also here, our PIs, our microscopy. I don't know if we have anyone here from histology, as well as our judges to come up, and we'll take a nice group picture. But before we do that, I just want to say thank you. I want to thank you all for coming around to our event. I'm sure right now you have, in fact, worked up an appetite for dessert, but also hopefully flagged a few projects that you would like to learn more about. And if nothing else, I hope you've expanded your view of what you've learned, of what's happening here at MIT, and integrated some new ideas into your thinking, or at least generated some new understanding about the current research happening at the Koch Institute and beyond. We have, in fact, made a lot of progress since our building opened in the last 5 years, but we're going to keep on glowing. We'll keep on with daring eats of engineering and robust biological models. So on that note, you are all welcome to revisit the images and talk with our presenters in the galleries. Please don't be nervous. Just find someone you want to talk to and feel free to put them on the spot. Thank you. It's been a pleasure.